And we're back. Welcome back from the weekend. Today we will continue on with our discussion about features, and we will focus today on certain categories of appearance-based features. But before we begin, some administrative bits. The reading by next week, 15th of September, which is a week from today, being Tuesday the 8th. There are 3 papers that are on the blackboard: automatic fruit recognition from natural images using color and texture, a detection method for apple fruits based on color and shape features, and color and shape recognition, pages 5 through 16. Now, for the first 2, certainly you will not understand everything that's in these papers. That's okay. The focus will be on the feature representation for those first 2. And this third one, that should be very accessible to you. And they're all on the blackboard as we speak. Okay, so last time we talked about the pattern recognition system architecture, the pipeline, as it were. And last time we focused on the sensor. Today we're going to focus on feature generation. And feature generation, we described in our first module the processes and decisions about what form or representation is assumed for each object within the data that are obtained from the sensors. We talked about the intensity surface representation, and now we will continue on with what kind of things we can do with this intensity surface representation. So let's begin with a motivating application. And this is image clustering, and you might be wondering, well, why is image clustering useful? Now, clustering is the grouping together of things that are deemed to be similar by some metric. And so in the old days, when I was a kid, young kid, probably 5 years old to 12 years old, predominantly the way you took photos was with a film camera. And so this is a Nikon, which is a pretty good camera back then, still is now. You have a roll of film, and you would buy this film for about $5, and you would take your pictures, and as you took the pictures, it would pull this film out of the canister, and then before you went to get it developed, you would rewind this so that the film would come back inside the canister where it was protected from the elements, and then you would bring it in for film processing. And so this film processing was about, at the time, $7.99 or so for a roll of 24 exposures. And then you get your processing, look at your photos, and then you'd see all of your shots, and then you would store some of these photos in various collections of photo albums. In fact, I have photo albums from when I was a kid growing up, before digital photography was very popular or even technologically available. And so the practice was you kind of hold your shot, right? I had a small 35 millimeter camera that I used to bring around with me all the time. And if you saw something interesting, you'd take a picture, but you wouldn't just arbitrarily start pointing and shooting, because this process was often very expensive. Now, the film was about $5. The processing for developing was about $7.99. You get your prints, and then you buy a photo album for about $10 to $20. So that entire prospect was relatively expensive. Then what happened? The proliferation of digital cameras, not even just digital cameras as a device in and of itself, specifically digital cameras in cell phones. Now, why is this useful? Well, because it was almost free, as it were, the cost of taking an image, right? You didn't have to do all that processing and pay the $7.99 and pay $5 for a new roll of film. You just take your photograph and build up enormous libraries. Even individuals are amassing photo libraries over years of tens of thousands of images. So why is it useful to do image clustering? So suppose you built up a huge photo library. In this particular case, we're just depicting images of different types of dogs. And you want to perform this clustering, so you separate them into different subgroups associated based on the content of these images. And then, of course, you go through after the fact and analyze these groupings and give them a name. So you might call the upper group the yellow Labrador Retriever, yellow lab. Maybe the pugs are this other group, and you name German Shepherds the other grouping. So you go back in after the fact, after the system or the algorithm has grouped together images from your library, based on similar content in these images, and you go back in and you, the human, gives them a label, gives them a name. So you might be wondering, gosh, well, if I wanted to do this, I don't really have a photo library. Where do I get a large number of images? Well, from something called computer vision databases. Now, these computer vision databases are often made available by the vision research community in order to share sets of data that enable others to reproduce results or to benchmark the performance of their algorithms. And so this lowers the barrier of entry for doing research, and it also facilitates solutions to important problems. So if you have an important problem, you create a computer vision database and you put it out there and say, hey, here's this great database, I have this problem, and people will pick it up and use it to see if they can solve that particular problem. And so here's one, face recognition. And for face recognition, oftentimes research is done under ideal conditions. And if you can see the face image of this person, this is from a face database, it's usually, this was certainly taken in the studio, and it's under perfect lighting conditions. So the light is ambient, fills the room, and you'll notice there's no sort of shiny reflection spots, and it's very well lit all around the subject. And the background is a matte color that does not take away from the content of the image per se. So now, for example, you might look at various pixels in the image as being good descriptors of what it means to be a face in this particular case. And not only do you look at the patches or regions of image that have certain pixel content or image content or values, you might look at the relationship spatially between regions in the image. And so this wireframe represents, these edges represent relationships between patches and various intersection points in the image. And so there's another database that has become pretty popular, it's very popular, called Labeled Faces in the Wild. It was done at UMass Amherst. And Labeled Faces in the Wild went out and solved the problem, said, okay, great. You have these image databases, these are taken in perfect studios, but what about under ideal conditions? But what about when face images for various computer vision algorithms, those faces occur out and about in society, so-called in the wild? And so if you notice here, the background is not ideal. So here we have a picture of B.B. King, and you'll notice there's some white in the background and some blue, and that's a fairly matte background. But you see in this image, this last image, you'll see there are other people in the image, so there are multiple faces. And so it's a harder problem when you're doing face detection and looking for particular faces to be able to identify the face that's particularly of interest, or if there's other background like trees and other objects depicted in the image content. And so one example image database that's been very popular is the Caltech 256 database, and there's the URL to get it. It's a total of 30,607 different images that are drawn from 256 categories. And so if you're looking for a large number of pictures of different type of objects, 256 different types of objects, the Caltech 256 database is a choice that's available for free, and it's used for very many pieces of computer vision algorithms research. And so this is an example from the Caltech database. Has images of dogs of different type, 102, 84 images from the giraffe category, and 102 images from the bear category. So let's take a look at so-called appearance-based features. Now, for appearance-based features, appearance is color and shape together, where color looks at the distribution of red, green, and blue components that make up the color channels for an RGB image. And it doesn't have to be just RGB, but we are focusing, for the sake of this discussion, on RGB representations. And then we also have shape. We talked about edges in the intensity surface, and we looked at the edge map, which is the result of taking the derivative with respect to the x scan line, the partial with respect to the y scan line, and doing the inverse tangent to get the edge orientation. So the shape is represented as a distribution of edge magnitude as well as or edge angle. And so if you look at the following two images, we make a few observations. Well, on the lower left-hand side, you have an image depicting a building. sort of like a corporate office park. In this building, if you take a look at the color, well, there's a lot of gray, and you have blue sky. Certainly, you have this dark charcoal gray for the street, and then you have the green for the grass and the landscaping, the shrubbery, and the trees. If you look on the right, this image of a natural scene, the sunflowers, a field full of sunflowers, you have lighter yellows, darker yellows, a little bit of browns. You have green, certainly a different green from the landscaping for the building. And then you have this blue sky. Well, the blue is a little bit different from the blue for the building. It's laden with clouds. And you also have some trees a little bit in the background on the right-hand side. Now, if you look at the shape, the edge distribution, well, this building, it has a lot of horizontal lines and it has a lot of vertical lines. And you'll notice here for these windows here, you have a vertical line and you have horizontal lines. So if you were to take the distribution of edge orientations for the building image, you're going to get high peaks in that distribution corresponding to 180 degrees and 90 degrees, and because those are very frequent occurrences for edges in this image. Conversely, if we look at the image for the sunflowers, well, sunflowers are round, and if we were to kind of trace straight edges for the sunflower, right, you're going to get a lot of representation in your distribution for all sorts of angles from 0 to 180 represent the orientation of edges. And so you're going to get more uniform distribution in the edge orientations for the sunflower picture than you will. It'll be very peaked, mostly bimodal, for the distribution representing edge orientations for the building. Okay, so we take our color and we extract the red, green, and blue color channels, and that gives us our color channels separately. We compute histograms of these color channels, and we assume, in this example, we're going to bin all of the 0 to 255 alternatives we have for the red channel values, the green channel values, and the blue channel values, and we're going to crunch them down into one of 10 bins. And so we do that for each one of the color channels, red, green, and blue, for the building. And likewise, we do the same thing, red, green, and blue, for the sunflower image. So then we take these bins and we collect them. And these bins will have counts representing the different color values at different saturations for red, green, and blue, 10 different levels into which we quantize our color values. And we collect them together to form a single vector. So in this case, for each image, both the building image and the sunflower image, we're going to form a single 30 vector. 10 of those positions in the 30 vector are from the red color channel, 10 from the green color channel, and the remaining 10 from the histogram bins for the blue color channel. So we do this both for the building image as well as the sunflower image. So we get a single 30 vector that collectively, each one of the positions describes the color content for the respective images. So now we do the same thing similarly with the edge angles. We compute our convolution mask and we compute our partial derivative with respect to x and with respect to y, perform the inverse tangent of the y response over the x response, and that gives us all the edges, or the orientations of the edges at every pixel in the image. So then we take those pixels and we calculate a histogram over all those edge angles. Unless we use 10 bins, we get this 10 vector representing the bin counts. So we're quantizing all of those edge orientations from 0 to 180 degrees into 10 bins. And we take those counts and we treat those as a 10 vector. And those feature vector for that is 10 positions wide, where each position contains the bin count for each one of those quantized edge angles in the original image. So we do this for both the building image and the sunflower image, resulting in a 10 vector for representing the shape or edge orientations for the building, as well as the shape for the edge orientations in the sunflower image. So now we said appearance is color and shape. So we take our 30 vector in each case for color, and we concatenate that with the 10 vector for shape, and this gives us both feature and shape rich description in a 40 vector for all of the content in the image. And so together, these feature vectors for color distribution and shape distribution or edge orientation gives us a better representation of the content that's in the image. Okay, so we can abstract this mathematically in vector form. Let's define vector bolded. That's vector notation. I don't draw the little arrow above the variable. I do it in bold because that's the style or the approach that I will take, which is reflected in the computer science literature. So we have vector x, and it's the ith x vector, and it represents the feature or the appearance-based feature description for the ith image. And it doesn't matter what I is, you have some number of images. The ith vector is the feature representation for the content of the ith image. And so each position in this ith vector, each one is a 40 vector in this example. Position 1 represents the first feature. Position 2, xi comma 2, is the second vector, up to and including xi 40. Now, each one of these positions, those are the features. The first 30 positions are the color descriptors, and the last 10 positions are the shape descriptors. And so we can describe this image mathematically, in this case, as a 40 vector. And this 40 vector, you can actually plot as a point in 40-dimensional feature space, as it's called. And so here we have the ith image. We have the first feature vector through the 40th feature for this particular 40 vector representation. So an image is represented as a vector in 40 dimensions, which is also, as I said before, a point in 40-dimensional feature space. So let's go to an example in 2 dimensions because I want to make a point about now how you would compare 2 images based on their content. And so again, we have the building image on the left, and we have the sunflower image on the right. Now let's assume we had a 2-dimensional representation, and it doesn't matter what the features are per se. You can imagine just one color feature, which is the average of all the color values across red, green, and blue, and then we have the average edge orientation for each of the images. So it doesn't matter how we've computed these features, these 2 vectors. What matters is that we have some vector representation, and I'm choosing 2 just because it's easy to talk about on a screen. So we have our feature vector in 2 space, and therefore it is called a 2-dimensional feature representation. And we have, for the first feature vector representing our building image, we have x11 and x12 for the first feature, x11, and the second feature, x12. Likewise, we have similarly for the sunflower picture, that's our second picture, so we represent that vector in 2 space as x2. It has 2 features, the first feature x21 and the second feature x22. So now let's define a coordinate system, and we're going to now represent each one of our images, the content, as a point in 2 dimensions. So we have 2 axes here, coordinate axes. We have the first axis, the x1 hat direction, and the x2 hat direction. And so now we can literally plot each one of these images as a point in 2-dimensional feature space. So here's the first image. This is our building, represents our building. And here's the second image that represents, for example, our sunflowers. And so now if we take a look at the projection of x1, that first feature vector, along the horizontal axis, that projection is going to be the first feature value, x11. Likewise, if we project it along the second axis, x2 hat direction, that projection of x1 along the x2 hat axis, in the x2 hat direction on the second axis, that's going to be our second feature, x12. Likewise, we do the same thing with our sunflower. It has a value for the first feature, which is its projection along the first axis, and its value x21, and it has its projection along the second axis, which is the second feature in this representation, value x22. And so you can imagine if we have each vector is in 40 dimensions instead of 2 dimensions, each one could be represented as a point in 40-dimensional feature space. So now we have our representation in feature space of each one of the images. So if we had some number of buildings, we'd have a number of dots. If we have some number of sunflower pictures, we'd have another set of dots, 1 point in feature space for each instance of our image. Okay, so now we treat these images as points in 2 dimensions. We can also treat them as vectors in 2 dimensions. As vectors, of course, we draw the butt of that vector at origin, and we have the arrow for that vector ending at the coordinate in feature space for each one of these vectors. Okay, so as vectors, certainly we can project them along the horizontal axis, the x1 axis, or the x2 hat axis. And again, these projections are their feature values for the respective points treated as vectors. Okay, so one of the questions one might reasonably want to ask is how different are these images, or how similar to one another are they? Now, this is why this representation in feature space is very important. So all we do is we just measure this distance between the 2 points, this geometric distance. And now we have the tools of Euclidean geometry to help us. And so it's just literally the L2 norm between these 2 points in vector space. And this gives us a measure of the distance or similarity between the content of the image x1 represented in feature space and image x2, or second image represented in feature space. So given that, if we wanted to group together images based on the appearance, the color, and shape, we can now take points representing a set of images and now compare the distances and group together those that are closer to one another and forming groups. And now we go back in and examine the results of things that were grouped together. All right, so likewise, treated as vectors, we can use the so-called cosine distance, which are between the 2 vectors, as another measurement of the similarity between 2 images. And so the cosine distance falls out of the dot product, A times B is equal to magnitude A times magnitude B cosine of the angle between them. So we divide each side of equality by the magnitudes, and we're left with a definition for cosine of the angle between them. That is the so-called cosine distance, which is another way of measuring the difference or similarity between 2 images. And here we're treating the points as vectors in 2 dimensions. Okay, so we talk about the basic representation, but this can get even more sophisticated. So we talked about the 40 vector, the first 30 positions representing the color derived from the histograms of the red, green, and blue channel. And we talked about the last 10 positions representing the distribution over the edge orientations representing our shape. And so we depicted this in a mathematical abstraction of a 40 vector, so a point in 40-dimensional feature space. So let's take this graph. We have our 30 vector representing color. Now, one of the things you might ask was, gosh, well, what if you didn't use 10 bins? What if you increased the number of bins? What happens? So in this particular example, I've increased the number of bins from 10 bins to 20 bins. So I've doubled the number of bins. Well, what happens to the representation if you now collect this? You have 20 bins contributed by the red, the green, and the blue for a total of 60 vector. So now the color component for this image is no longer the 30 vector that we had before, assuming 10 bins per color channel. Now we have a 60 vector. We've now increased the contribution, the dimensionality of the color description. So what does more bins do? Well, more bins means that the range of pixel values represented in a single bin is going to be smaller, because remember, we can have 255 different saturations for the red, saturations for the blue, and saturations for the green. If we narrow the number of bins, that means the range of saturations is going to be fewer, represented by a single bin. We're quantizing into a larger number of categories or levels, and therefore the range of saturations or range of colors represented by a single bin is going to be a smaller range. And so this means that you're going to represent more distinguished, smaller variations in color. So, for example, if you're measuring the color of a giraffe or representing the color of a giraffe with more bins, that means you're going to distinguish among different giraffe pigmentations. So a giraffe that's light brown, dark brown, and medium brown might appear as different vector values if you're dealing with a larger number of bins, because these smaller variations are going to result in different vector position numbers, different feature values. So conversely, what if you had fewer bins? Now, originally we talked about 10 bins per histogram over each one of the color channels, forming a total of a 30 vector for the color portion of your representation. If we reduce that to 5 bins per histogram, if you collect those bins together, it results in a 15 vector instead of a 30 vector. So it's a smaller representation in terms of the dimensionality of that component of the feature vector representing the color content of the image. And so what happens if you have fewer bins? Well, if you have fewer bins, effectively, again, the range of 255 values is now quantized into 5 levels instead of 10 levels. And so that means each bin represents a broader set of values. And so you're going to collapse a broader range of pigmentations for the giraffe example into a single representation. So that means differences in pigmentation for the giraffe that might be dark brown, light brown, or medium brown are just categorized as brown. And so with a smaller number of bins, you get more fine-grained resolution for representation. For a smaller number of bins, you get more coarse-grained, representation, more coarse-grained resolution in your representation. For a larger number of bins, you get more fine-grained resolution in your representation. And so for the fewer bins, that means fewer differentiation on the color. So everything that's light brown, dark brown, or medium brown would just be brown. Okay. So let's take a look at the edge angle, right? In similar fashion, we started with 10 bins for the histogram over the edge orientations or the edge map computed using the inverse tangent over the y gradient and the x gradient in the image. And so if we use a larger number of bins, what happens? You distinguish smaller variations in edge angles. So things maybe that are 35 degrees, 45 degrees, 55 degrees are all different. So you can distinguish these more fine-grained variations in edge angle for the edge content for shape for an image. If you have fewer bins, you're collapsing edge angles. So things that might be 35 degrees, 45 degrees, 55 degrees will all be in one category called slanted. You have a slanted edge versus a 35-degree edge. Okay, so again, if you have more bins, that's more fine-grained resolution and representation of different edge angles. If you have fewer bins, that's more coarse-grained, so a bunch of different edge angles will now be collapsed into one category. Might call it slanted edges. So let's take a look at some objects, and we'll talk about this idea about what is an efficacious number of bins, because the bin representation that you use can make or break the performance of your algorithm. If you choose poorly based on the type of images you have, you're going to get poor performance. So let's take a look. We have a picture of a baseball, and we have a picture of the gabling on a roof. And so here I'm going to draw some tangent lines to points along the edges of each one of these images. Now, for a baseball is round, and round things tend to have a more uniform distribution over the edge angles. So in the case of a baseball, that uniformity is more obvious if you have a larger number of bins, because you're going to get basically a similar or same amount of counts across a larger number of bins. With a gabled surface for a house or on the architecture, if you notice here, I have a slanted line that's about 45 degrees, and this comes into contact or intersects with the other slanted line that's about 45 degrees off of 180. So we have 45 degrees off of 0, 45 degrees off of 180. Now, engineered objects, human engineered objects, tend to have straight edges. And whenever you have straight edges in a distribution of edge orientations, that's represented as a very highly peaked bimodal or trimodal, small number of modes or small number of peaks in the resulting edge distribution. And so that means or translates in the image content to having a handful of dominant edge angles. And that's certainly apparent here in this house. You have the edge here for one side of the gable, edge here for the other. You have a horizontal edge for that lip of the house. Same thing for where the shingles meet. Likewise, the edge of this roof is the dominant edge. And so you might have fewer bins for these human engineered objects in order to suitably represent them, because everything else would just be noise. All right, so we can also have more sophistication in considering the difference between global and local representation. And so in our original conversation, we took the entire image for shape and we computed the gradients and expressed the inverse tangent, gave us the edge orientations over the whole image. But when we did this, we have no representation in this 10 vector for the relative position. And if we look at the image, we see, well, gosh, well, we have the eye, that's one shape, and some distance away from that eye is a similar shape for the other eye. Likewise for the ears. There's some symmetry here. For these little horn things, bumps on the top of the head, there's some symmetry there. There's an angle here, and then there's the converse angle on the other side, reflected about the vertical axis. So there's no notion of relative relationship. So one of the things we can do is we take that image and compute what's called a local shape descriptor. And in doing so, we're trying to capture the relative relationship between patches or regions within this image. So one way we can do this is we tessellate or divide the image into cells, and then for each cell, we compute the edge orientation just for the edge points within that particular cell. And so here we have a 4x4 tessellation of our image for a total of 16 cells. And then if we compute a 10 vector for our histogram, so we compute the histogram using 10 bins for the edges from each cell. So we do this, and that gives us 10 bins or 10 values or features from each of these 16 cells. And so we put that all together, that gives us 16 times 10, which is a 160 vector, and that 160 vector represents the edge orientations for one feature, one aspect, a local patch for one cell in the image. And so if we look at this vector, it's literally capturing the fact, for example, for this cell, for the ear, it's related to this cell on the other extreme for the other ear. And so in doing so, we're not only capturing the edge orientation, we're capturing the edge orientation or description of edge orientations or as distributions for local patches of image, and simply by ordering these cells in some fashion, maybe the first cell, the second cell, the third cell, and we go in some regular order for all images, we're now representing not only just the content of the edges, but the relationship between the edges in different parts of the image. And so now we can use this 160 vector as a more rich local shape. We take the parts of the image and say, this is the shape in this part relative to the other part. And so this gives us the added descriptor of relative relationships between patches or regions of your image. Okay, so let's take this idea and we're going to talk about HOG, which is Histogram of Oriented Gradients. And so what are HOG features? In HOG features, we take this idea of local shape and we concatenate them in a very clever way and to give us a very powerful descriptor of shape. So we start out with an image and we divide it into cells. And a cell is one of these small green squares. And we define a block, one of these red squares, as being m by n cells. And so here we have a block, and let me write the name block, O-C-K. Let me switch from my mouse to my pen so I get a little bit better control over what I'm writing. And a block is going to be m cells by n cells. So a block is composed of m by n cells. In this particular case, if we look at the red block is just 2 by 2. It's 2 rows of cells by 2 columns of cells. So we have our image divided into cells. We define a block as m by n cells. And so now we take the block and we imagine this block superimposed over our image, and we slide this block across the image, both across, and when we get to the end, we start at the left-hand side again, bring it down, and keep doing that operation. And so we take this block, and you'll notice in one position here, block 1, that's the block at the first position, we slide this over one cell at a time. And you'll notice the second block, the blue block, that represents the block at the next position. Now, when we slid this over, one of the observations that you'll make is that the block in the first position, it overlaps 50% with the block in the second position. So we take this block and we can move it so that it's non-overlapping or it is overlapping by some percentage. And for each position that the block is in, we compute the edge gradients for each of the cells in the block. So if we're in this first position, we have our image here, and our block at time t, or position, the first position, it's in that upper left-hand corner. That's the first position. In the next instant, when we move the block to the next position, well, it overlaps where the block was in the first position by 50%, and the block is now in this next position. And so for each position of the block, we're going to compute the edge gradients for each cell. So we compute all the edge gradients, theta, for each one of the cells for the block in the first position, and then likewise we do the same thing for each of the cells for the block in the second position. Now, of course, you'll notice that the block in the first position overlaps the block in the second position by 50%. So that means we're going to maintain some kind of consistency because the image only changes so much if these blocks overlap with one another. And so we compute all of the edge gradients for every pixel in every cell for this block. And so we take all of those edge gradients now and we combine them through a voting mechanism. And so each cell within a block is going to contribute some number of edge measurements, some number of theta, and we're going to take that number of theta, and each cell in the block is going to use its edge orientations to vote, and it's going to vote in a so-called quantizing histogram. And so this particular histogram is depicted here, and this histogram sweeps from values, and these represent edge angles from 0 to 180 degrees. And you notice here, each bin of the histogram is represented as each one of these wedges, and each bin spans a total of 20 degrees. So if an edge has an orientation between 0 and 20, it's going to go in the first bin. If it's between 20 and 40, it's going to go in the second bin, and so forth up to that last bin. So we have a total of 1, 2, 3, 4, 5, 6, 7, 8, 9 bins. 9 times 20 degrees is a total span of 180. So each histogram is going to be run over every single cell. And so for a single block in a position, we end up with a histogram representing the edge orientations for the first cell, a histogram representing the edge orientations for the second cell, a histogram representing the edge orientations for the third cell, and a histogram representing the edge orientations for the last cell. And so now what happens is that for each of the bins, the first bin through the 9th bin, for each one of these histograms, they're going to do a vote. And whichever bin is the greatest, so here's bin 1, bin 1, bin 1, and bin 1, whichever bin for the first cell, the second cell, the third cell, or the fourth cell has the largest bin count, that's going to be the winner of the vote. And so that's going to define the winning edge orientation as well as the edge magnitude. And so through this voting mechanism, you're getting somewhat of robustness, because what's going to be representative of the dominant edge orientation is going to be that particular bin's edge contribution if it has the biggest edge count. Okay, so given this voting mechanism, we get two things. We get a semblance of robustness within a region of image, and we also get consistency because this block, as we move it, we can overlap it and keep consistency from one position to the next. Moreover, as we move it, by putting together all of these resulting votes from these 4 cell histograms, we get this notion of local features, because essentially we're doing a vote for this position, a vote for the next position, a vote for the next position, a vote for the next position. So we're getting relative edge information for regions of image that maintain consistency between one patch of the image and another patch. And so if we do a vote for this position and then a vote for that position, we know that we have certain edge orientations here and other edge orientations in some relationship to that location within a patch of image. And so we take each one of these winning histograms and we concatenate these 9 bin values resulting from the vote, and we get for each position, we get such a histogram, and we concatenate them out as we slide the block across the image, and the result is our entire feature vector. So we have our feature vector, and it's composed of the voted histogram bin for each block position. Okay, so we concatenate all of these histograms into a single feature vector. So as you can imagine, if you have a lot of block positions, you're going to have a really big feature space representation for the content of that image. If you have fewer number of block positions, you're going to have a smaller feature vector for the representation of that image. But the bad news is, if you have a big feature representation, well, that's nice. You're going to have very high level of granularity and a lot of spatial representations. If you have a small number of block positions, you're going to have more coarse-grained granularity, but you're going to have a smaller representation, and your relative relationship is going to be between regions of the image that are farther away from one another because those block positions are fewer. Okay, so this is an example of hog features, and it's called histograms of oriented gradients. It's oriented because you're looking at these various orientations, and the winning bin is the one that gives you the dominant edge orientation. And here we have the top few edge orientations for the different bins positions. And in this image, we have the original grayscale image. So we have a person walking along a dock near a waterway. And then we also have a person standing at the beach with the arms extended wearing this red jacket. Now, it just so happens that the blocks here are non-overlapping, right? And so we have zero overlap between the blocks. And in this resulting image, we have, you can see the position of the blocks, and plotted are lines that represent the top few dominant edges. And so if you look at where this person is walking, they have some ripples on the water. And you'll notice here the dominant top few orientations are roughly aligned with the edges for the ripples on the water. And we get that all along for the ripples of the water until we notice the shoulder of the person walking, and you'll notice the arm kind of extended out at some angle. Well, we capture those edges. We capture that part of the arm, that part of the arm, and that coincides with the top few dominant edge orientations or dominant edges align perfectly with that arm that's extended. And then if you see this side of the person, well, we see that represented here with those dominant edges that are kind of a little bit off center, but mostly vertical. And then we also see that line here, the jacket makes that edge with the person. Here's that line. And then we see the other end of the jacket. Here's that line, right? And it coincides perfectly. We see the person's left-hand leg, and you'll notice that part of his hip is slanted. We get that part of the hip, it's slanted a little bit. We see those slants. And then we have the shin here, right? There's the shin. And there's the other side of the person's leg. There's the other side. And then we also get that slope here of that person's backside. There's the slope here. And it matches perfectly. We even see the alignment of this dock, the person that's walking along. They see the edges. We notice those edges here that align perfectly with the dock. Let's cast our attention to the other image. We see this person standing up, and we see, let's see, the person's right-hand thigh. Well, there's the right-hand thigh. There's the edges there. And we see the crease between the person's legs. Here's the crease. Here's the left-hand side of the thigh, left-hand side of the thigh. So let's see. Here is the right arm. Well, there's the edge for the right arm. Here's the edge for the left arm. You notice here the flare in the jacket that comes out. There's a little bit of flare that comes out here and here. And then the person's head. You see here the person's head there. And you see there a little bit the person's head. Okay, so these links talk about hog features, but this is just an example of a representation that trades off local versus global. And in this trade-off, you can represent, because it represents the sliding of a block, which is m by n cells wide, you capture the spatial relationship between local regions of image, and then you also capture some aspect of global representation because you're doing this histogram voting, so you're abstracting over all the low-level details at each location within the image. Okay, so these papers, please take a look at them, and it will tell you a little bit more about histogram of oriented Gaussian or histogram of oriented gradient features and how they work. So in MATLAB, actually, we have API calls in the image processing computer vision toolbox that will actually extract hog features for you. And so in this API, you decide what the cell size is, you decide what the block size is, you decide the overlap between subsequent speeds of the block, and not only does this give you representation, specificity, level of granularity in the representation, in terms of how descriptive or how distinctive it is among minor variations, it also results in a speed trade-off as well. And so if you have many more block positions, so the overlap is very large from one block position to the next, it's going to run more slowly because it takes time to slide this block across and do all those computations of the binning and the voting of the resultant histograms. Okay, so with that, that's all I had for today, and we will end there. Again, the reading by September 15th, which is a week from today, is please make sure you are paying attention. These are on the blackboard. Please make sure you're paying attention, special attention to the appearance-based features, color, shape, and one of them also talks about texture. Okay, so with that, we'll end there. And as usual, please stay healthy and stay safe, and I will see you all on Thursday.